Assalamualaikum everyone. Today we are going to discuss about the bacterial species identification on MALDI biotyper. MALDI-TOF is basically used to sequence proteins to identify microorganisms and to carry out several biochemical reactions in a single day. These are some of the reagents which are used in the process of MALDI biotyper. This is the kit of MALDI biotyper. I will put out the plate out of this kit, and first of all I will clean it because this MALDI plate is already being used. So I'll spray the 70% ethanol solution on the MALDI plate and will clean it. 70% ethanol will be flooded on the both sides of the MALDI plate for the perfect cleaning. I'll clean 4 plates because I have 4 plates. Cleaning process is being done with the autoclaved cotton swab, and I'll dip this cotton swab into the ethanol and will softly clean all the wells of MALDI plate. You cannot push it hardly to prevent the scratches, because if there will be any scratch, the detector will be unable to detect the organism. You will softly swab the cotton swab on your MALDI plate. This is the TFA solution which is being made in the laboratory. Now it's time to pipette this TFA solution on the MALDI plate for further cleaning. You will not flood the TFA solution on your MALDI plate. Rather than that, you will pipette it down because it is already made in a very small quantity. And again, you have to clean the plate with the cotton swab. Precautionary measures are still the same, that you cannot push harshly. The whole procedure is being taken place in the safety cabinet, either BSL-2 or BSL-3 level, depending upon the organism you have used. Now it's time to dry your plates with the soft tissue paper. You cannot use a hard tissue paper in this step. So I am slowly tapping the MALDI plates with the tissue paper. Then I will dip them again with the at 70% ethanol and will again dry them. So all this process is performed on all the 4 plates step by step. As you can see that I am wrapping the plates in soft tissue paper. This is how a MALDI plate looks like. You can see the labeling from 1 to 12 in the horizontal direction and A to H in the vertical direction. You have to prepare the key beforehand for the spotting process. You will run 6 controls, 2 of bacterial strain standard, 2 positive control and 2 negative control. There are 2 types of standard which are run in the MALDI process, the one which have HCCA matrix and the one which do not have HCCA matrix. When the spotting is being done, you spot the 1 microliter of HCCA matrix on each well to fix your strain. So if your standard already have matrix, you will not add it later on. This is the HCCA matrix which is being used in the fixing process. Now it's time to put the MALDI plate into the MALDI biotyper machine, and this is how it looks like from the outside, and it will send you a report in this form. The score above 2, which is labeled as green, is the exact identification of your organism. The score below 2 and in between the range of 1.8 to 2, which is labeled yellow, indicates that you have to redo the experiment for the further confirmation of your organism. And the score below 1.7 indicates no organism identification, and it is labeled as red. If there will be any scratch on your MALDI plate, the biotyper will be unable to identify your organism as well. So be careful while cleaning, and this is another representation of your results. The green boxes indicate the exact identification of your organism, the yellow indicates that you have to redo the experiment, and the red boxes indicates that no organism is being identified. As you can see that the laser light is capturing the specimen from different angles so that the perfect identification can take place. And you also get the report on the side of your screen in this form, as you can see. Thank you so much for watching this video, and if you have any question you can ask.